And we will start with the first talk by Professor Aaron Dinner from University of Chicago, who is going to talk about estimating commuters and relative currents through dynamic approximation. So, Professor Dinner, you have 30 minutes for talk and 5 minutes for question answer. So you can start your talk now.
So let me begin by thanking the organizers. So far, it's been a really great meeting, and I'm only sorry that we're not actually meeting in person. So what I want to talk to you about today are some data-driven approaches that we've developed for computing kinetic statistics. And this is work from a number of my former and current students, and it's a long-standing part of a long-standing collaboration with Jonathan Ware and his student Rob Weber at New York University. Okay, so, oops, it won't let me move. Okay, so kinetics are important, and just to give you one example, a system that I'm going to show you some results for later, there's tremendous interest in being able to design insulin therapies with particular pharmacokinetics. So you might want a fast-acting insulin that you could take right before a meal, or that you could put in a pump that somebody would wear and would be continuously monitoring the glucose levels in the blood. You might want a slow-acting insulin so that you only have to take one shot during a day. So insulin, the protein hormone, it takes a variety of oligomeric forms. When it's in vesicles in the pancreas and then secreted, or in a therapeutic formulation, it's typically in a hexameric form. It then dissociates to make dimeric form. That's the predominant form in the blood. And then, as Biman Bagchi mentioned, what was my morning, I guess, yesterday evening, for some of you, then this dimer has to dissociate to a monomer in order to bind to the cell receptor. Okay, so you can see that actually the different transitions between oligomeric forms have different kinetics. And so we would like to be able to understand those kinetics and the mechanisms underlying them, because each of them is a point where one could actually manipulate the system to try to get better overall kinetics, okay? So that's just a motivating example. I'm sure many of you can think of your own examples of interest. So I don't need to tell this audience that because there's a separation in timescales between the time step of a numerical integrator and events of interest, right? In insulin, you saw that that graph had an axis in hours. Because of that separation in timescales, you can't simulate these dynamics directly, and you have to do something else if you want to get at the kinetics. And we've heard about a variety of different enhanced sampling methods already in the meeting. The one that I want to talk about briefly, because it's going to set the stage for what I'm going to tell you about, are Markov state models. So the idea is that you have some dynamics, however generated. We'll talk a little bit about that in a bit. And then you defined states. You bin the trajectories into those states, and then you determine the state-to-state transition probabilities. Once you have that transition matrix, then in principle, at least at the coarse-grained resolution, you can calculate the full evolution of the system, and so any quantity that you might want, okay? In practice, what is often done is simply to look at the eigenvalues of the transition matrix, because those provide information about the slowest relaxation timescales. But there's an issue with that, which is that the process of interest might not be actually the slowest one. Maybe I'm interested in how, say, a hexamer comes apart into a dimer, but actually the slowest relaxation timescales are the unfolding of the monomer, for example. An additional issue is if I don't specify a reaction a priori, then actually the computation of the statistics that I would like, so things like committers, mean first passage times, rates, those will actually be biased by using the transition matrix that I would construct just from all of the states without knowledge of the reactant and product states. So what I want to tell you about is an approach that we've been developing the last couple of years called the dynamical Galerkin approximation. And what it does is it shifts the focus to actually directly solving for kinetic statistics. The way that it does that is cast those statistics as solutions of an operator equation, where that operator is a so-called transition operator, which tells us about the expectation, the evolution of the expectation of functions. So in some sense, the way you could think of this dynamical Galerkin approximation, or DGA, is as a generalization of Markov state models, but one where we've actually specified what is the reaction or process that we're interested in. And then we're going to be able to correctly account for the, in essence, reactant and product states when we construct the appropriate transition matrices and then solve these operator equations. Okay, so that's the basic idea. So to make this a little bit more concrete, one very important statistic that we will want is the committer. So Ron Elber introduced this statistic yesterday. It's the probability that I go to the product state before returning to the reactant state. So a committer of close to 0 would be part of the reactant state. A committer of close to 1 would be part of the product state. And then a committer of 1/2 would be a transition state. I have an equal likelihood of going forward to the product or back to the react. So in some sense, this committer is a perfect reaction coordinate. It's always telling us about what's the likelihood that we complete a stochastic reaction. Moreover, I'll show you that we can actually compute other statistics from it, like rates. But traditionally, this has been a very hard statistic to obtain, because traditionally what people have done is they've started with a configuration and then randomized momenta and then run new molecular dynamics simulations. And so then to evaluate the committer for a single configuration actually takes many more simulations. Okay. So what we're going to do instead is solve an operator equation for the committer. And that operator equation is T, Q equals to T. Ron actually had a version of this equation in his talk, just using different symbols. And then we have boundary conditions associated with that equation. There's 0 when we're in the reactant state and 1 when we're in the product state. Okay. I could write down similar kinds of equations for other kinetic statistics that I might want. The so-called backward committer, so that's the probability that I last came from the reactant, and actually the stationary distribution, so the potential of mean force in effect. If we have those 3 statistics, then we can actually compute the flux from the reactant to the product. Moreover, we can break that flux, which is tantamount to the rate, down into currents through different regions of space. And those tell us about what are the pathways that are contributing to the reaction. So these are the kinds of statistics and the kinds of equations that we want to solve. Okay, so the way we're going to solve those equations is that we're going to expand each statistic in a linear combination of basis functions. So we're going to have one that's going to account for the boundary conditions. And then we'll have a sum over ones that solve the homogeneous problem. That way we can add and subtract these phi functions without going out of the space. Okay, and then what we do is we proceed very similarly to what we would do if we were setting up the secular equation in a quantum mechanical problem. We take this basis expansion, we substitute it into the operator equation, we multiply by another basis function, and then we integrate to form inner products. And if we do that, we convert this operator equation into a linear algebra problem, just like quantum mechanics again. Okay, but here everything, of course, is classical stochastic. So these matrices C, they have elements that are of this form, phi, t, phi. And the issue is that in contrast to quantum mechanics, we don't know actually the form of T explicitly. If this was a simple diffusion, we could write down a differential operator, but actually here we have a finite time, and everything that is happening within our molecular dynamics simulation is going into that. And then we typically don't want to work in the full configuration space. We usually want to work in a somewhat reduced space. And so we have actually a kind of coarse graining where we don't know actually those dynamics a priori. Okay, so it might seem like that's a problem, but actually that's where the data comes in. So what we can do is we can actually estimate these matrix elements from molecular dynamics data. So we generate a database of trajectories, and then we pick up time points that are separated by a time tau, the lag time. And then we're just going to approximate the action of T on phi j by this difference of phi j at tau from minus phi j at 0. Then we just multiply by phi I, that's this part, and then we sum or we average, and that's the bracket. Okay, so basically from the statistics of trajectories, we can estimate these matrix elements. We can then solve the linear algebra problem, get the coefficients, and reconstruct the kinetic function of interest, like the commit. Now, something I didn't emphasize before is that when we form these inner products, it's an integral over a distribution, sampling distribution mu. And that can actually be an arbitrary distribution. So that's going to give us a lot of freedom to optimize the procedure. So it can be an enhanced sampling approach. For example, trajectory stratification. So that would encompass things like the milestoning that you saw yesterday, or non-equilibrium umbrella sampling, which is an approach that we've developed in parallel to RAN over the years. But you're going to see that actually it could be a less structured procedure. And so we can actually build up a database with a lot of flexibility. Okay. So I'm going to show you results for two systems. The first is really to validate the method, and then the second will be an application. So the first system is the well-studied trip cage mini protein. So this is a 20 residue designed sequence that's been studied by quite a few groups, both experimentally and computationally. You can see in this in this picture on the left that there are two folding pathways, one where the hydrophobic core forms first, and then this alpha helix, and one where this alpha helix forms first, and then the core. And we're going to make use of some very nice simulations from Peter Bullhouse some years ago. And what Peter did was transition path sampling, and then actually the difficult process of shooting in order to find, I think it was 12 transition state structures. So we can compare to those, okay? Okay, so the first part of our procedure is that we have to actually generate some data. And in our case, because we knew a lot about this system, what we did was we distributed initial points in the space of RMSD and then this 310 helix RMSD. So those are the blue dots here superimposed on the potential mean force that I'm going to calculate in a moment. And when we did that, actually, we found that we hadn't adequately sampled the unfolding of this alpha helix. And so we actually added a second set of simulations where now we're actually distributing points in the end-to-end distance so that we can ensure that we unfold that alpha helix. And so in total, what we've got are a little more than 1,000 trajectories with each one is 30 nanoseconds. And so we have about 30 microseconds of simulation. So it's actually really not very much when you think about it, okay? I'll show you we get really actually excellent results with it. Okay, so before we get to actual kinetics, let me show you the potential of mean force. So to apply the procedure, we have to choose a basis set. I could choose a basis set of indicator functions. That would be the sort of typical Markov state model states. Here, actually, what we're doing is we're choosing pairwise distances. So we have the freedom to choose a continuous function. We choose 153 pairwise distances between selective C alphas over the sequence. And then the basis functions have to satisfy the homogeneous boundary conditions. And so we multiply by the distance to states A and states B. So that takes those functions to 0. And then we do some PCA basically to, in essence, order and orthogonalize the basis. Okay. So once we do that, we can then make, in essence, free energy estimates for all of the states in our database, 6 million states in our database in this 153-dimensional space. And then we can project it down to different pairs of order parameters or collective variables to see which ones tell us the most. So here I'm just testing. I think it's 5 different order parameters. I'm going to show you our most of the results in the helix RMSD and the full RMSD because that's what Peter used in his study. But we actually find from our calculations that we get a little bit better description of the system with the helix RMSD and the 310 helix RMSD. And so actually, if you look at our published results, we have a lot in those coordinates. Okay, so now we're going to calculate the committer. Again, we do that calculation in 153 dimensions for 6 billion structures. So that's something that I never thought we'd be able to do. And then just for the purposes of visualization, we are projecting down to 2 dimensions and averaging, okay? And then we're comparing to Peter's results. You can see that the white regions here between the red and the blue, which correspond to committer equals a half, they correspond very well to the positions of his transition states, these red symbols. And furthermore, you can go in and you can look at the individual structures and compare them to his. And again, they look good. Okay, so we feel that that means the method is working. Then we can actually get a quantity that hasn't been computed previously, but is actually quite important in so-called transition path theory, and that's the reactive current. So this is, in essence, the local flux through a surface. And so in Peter's case, he showed just a couple pathways showing, in essence, the two pathways. We get a much more statistical view from the current. You can still see, in essence, the two pathways, but now actually from the color of these arrows, you can see that actually this one that kind of comes down and goes along the bottom is actually a much more populated pathway. And we can formalize that by dividing up this space and then integrating in order to calculate the flux through each of the two pathways. Okay, so we can make that quantitative. So overall, what the trip cage calculations are showing us is that the procedure is working. So now what I want to do is turn to insulin. I already basically introduced the problem. What it turns out is that actually in therapeutic formulations, there is often phenol included. It was first introduced as an antimicrobial, but it turns out that it actually stabilizes the hexamer, and so it increases the shelf life of therapeutic formulations. And so by understanding how phenol is released from the hexamer, we can potentially learn something about how we might make other molecules that would further stabilize the shelf life of a therapeutic formulation. Okay, so that's the motivation. Okay, just to orient you, like I said, Beeman introduced insulin in the previous session. He was looking at the dimer that you can see on the right. Here with this characteristic anti-parallel beta strand at the dimer interface. The hexamer you can think of as a trimer of dimers. So if I rotate the hexamer, I've got one dimer here, one dimer here, and one dimer here, okay? You can see that there are 6 phenols found in this structure. And it's a little bit difficult to appreciate from this representation, but the phenol is really buried. If I were to show you all the atoms, you wouldn't be able to see the phenol at all. So it's really a rare event for it to leave. Okay, so the first thing we have to do is we have to kind of do some exploratory calculations with a view towards being able to generate the initial points that we're going to run unbiased dynamics from. And so we did that through a kind of steered molecular dynamics method called adiabatic bias molecular dynamics. It gently ratchets the phenol out of the binding pocket. The first thing that those calculations show is that actually the escape channel opens quite easily. It happens quite frequently in our simulations, and that hadn't been seen before. And then if you count the closed and the open states as separate pathways, then we see 6 pathways of escape for the phenol. So these sort of van der Waals spheres are the phenol with many configurations superimposed. And so pathway 1 or pathway 1A, which is similar but has an open channel, that's the phenol coming up through these 2 green residues, which are so-called gatekeeper residues. The phenol has to go through the gate. Pathways 4 and 4A are the phenol going down through these 2 black gatekeeper residues. Pathway 2 is the phenol coming, in essence, between the 2 sets of gatekeeper residues, so avoiding, in essence, the gate. And then pathway 3 is actually quite different than the other pathways that I've mentioned. It's going out through the dimer interface. So it's a different direction, in essence, going back, as shown here in yellow. Okay, and some of these pathways, I should say, were seen previously. In particular, actually, Harish Vasith, who I think is attending the meeting, did some really beautiful simulations now actually quite some years ago, where they saw actually 3 of these pathways. And you know, we recover those, and then we see some additional ones, particularly ones associated with this opening of the escape channel. But in all of these cases, both the earlier simulations as well as the simulation that I'm showing you here, it's driven in some way, so they're biased. Okay, so what are the kind of questions that we want to ask? So just what are the unbiased dynamics? You know, what are the relative weights of the pathways in that case? Where are the transition states? What are the rates? And then how would mutations impact the mechanism? Okay, so the first thing we have to do is we have to generate some unbiased trajectories. So we pick points out of the ABMD simulations that are roughly evenly distributed in distance from the binding pocket. We run almost 700 40 nanosecond trajectories. So we get 28 microseconds of sampling. Again, actually not that much when you think about it. So it's pretty economical. We use a basis set similar to the one that I showed you before, except now we're working in a somewhat larger space of almost 300 pairwise distances. Okay. All right. So now we can apply our method. We can compute potential mean force, reactive current, and committer. I'm going to show you each one of those in turn. I'm going to show you them projected onto counts of contacts along pathway 1 and pathway 4, because that provides a pretty good separation of 4 of our pathways, actually 1 and 1A and 4 and 4A. Okay, so just to orient you, here at about 60-30 is phenol in the binding pocket. So it's in essence the crystal structure. These couple other minima, those are with the channel open and varying degrees of the phenol coming out. And then when the phenol is totally unbound, we're at 0, 0. There are no contacts, okay? All right, so that's the potential of mean force. We have minima at key states. Then we can calculate the currents along pathway 1. In essence, what happens is we loop out in this space following these arrows. We can look at the structures at kind of key landmarks along there and kind of see what's blocking the way for the phenol, and then eventually we loop back in to 0, 0. And I should say, with channel closed, you have a somewhat larger loop than with the channel open. And that's just because you're able to make more protein contacts when things are a little bit closer in. Okay, pathway 4, instead we go up and then down in this representation. And as we come sort of along the axis here, then a kind of interesting thing is that there's some melting of one of the helices of the A chain. Okay, so those are pathways 1 and 4. We can look at where the transition states are along those pathways. Those are marked by the purple lines in this committer plot. What's actually really interesting is that the transition states are quite late in the process. So they're once the phenol has already gotten past the gatekeeper residues, which I don't think I would have guessed before doing the simulation. Okay, so that's pathways 1 and 4. We'd like to look at pathways 2 and 3. In this space, those 2 pathways overlap. And so what we want to do is split the data into a third dimension. And the third dimension that we use is actually the committer, right? We can calculate it for all of the structures in our database, and it should be a perfect reaction coordinate, and so we might as well use it. And you can see actually it does a pretty good job of splitting pathways 2 and 3, the yellow and brown here. And then if I just summarize all the pathways over here on the left, now colored with the dots colored by the committer, what we can now do is we can make a cut horizontally in this space around committer equals a half. And that'll give us a plane, and we can then divide up where the different pathways are cutting through. And then we can integrate over each of these rectangles the reactive current, where we get the populations of the different pathways. So what we can see is that pathway 4A, that's with the channel open following the pathway 4. So the black gatekeepers, that's the most populated pathway, but all of the pathways have significant representation. It's about half and half open and closed channel, which again, I think would not be obvious until you can do the quantitative calculation. And so it's really kind of intrinsically a truly multi-pathway reaction. And so what I think that says is that really actually, traditional approaches that are based on a single transition state are not going to be successful. Okay, so very quickly, let me show you some results for some mutants. So we looked at 2 mutants, B13, which is a glutamate that sticks right into the middle of the hexamer. We removed a charge to make a glutamine, so you'd expect that to affect the channel opening. And A10, an isoleucine to valine mutation, so we took away a methyl group. It's one of the gatekeeper residues, so it should affect pathway 1. Okay, so here's B13. I'm just showing you the reactive current. We can calculate all the other statistics too. And from the difference, you can see that actually pathway 4 is really actually depleted. So you can see how things shift around here. A10, I'm just showing you the committer. And here's the difference on the right. Something to notice is that actually most of this plot is very close to 0. And so that actually speaks to the reproducibility of the calculation. Just by changing one residue, most things actually don't change. Completely new data set, new analysis. Where we do have a difference is where there's sort of this blue streak here. And so what that says is that the transition state is moving a little bit later. So it's favoring the bound form, disfavoring the unbound form. So that's really all I wanted to say about insulin. I did want to make one advertisement before finishing up. Something I didn't have time to talk about is some work that we did looking at the so-called variational approach to conformational dynamics. So that would include methods like TICA or VAMP nets. And what we show actually in this upper paper is that those methods are very sensitive to the choice of lag time. And we introduce a very simple fix, which is to integrate over the lag time. And we show that actually you maintain a variational principle. And then this, this picture and this lower reference is what we've done is we've taken now that integrated approach, IVAC, and applied it to understand the structural ensemble of the insulin monomer in collaboration with Andrei Tokmakov, and that's allowed us then to interpret some hydrogen exchange protection factor data from Mike Weiss, which had some difficult to interpret features. So if you're using these methods like TICA or VAMPnets, VAC, then I would encourage you to check this out. Okay, so what I tried to show you is that we've introduced this dynamical Galerkin approximation. Galerkin approximation, just a basis expansion for computing kinetic statistics. You can think of it as a generalization of Markov state models, but one where we're actually saying this is the reaction that we're interested in, and we're properly accounting for the reactant and product states when we compute these statistics like the committer or the reactive current. We validated it against a very well-characterized system, the trip cage, and then we've been applying it to looking at phenol release from the insulin hexamer. We've also done the dimer dissociation, although those calculations are not as well converged yet. And then what I showed you is that actually, because you're able to work in this sort of higher dimensional space, you can actually try out lots of combinations of order parameters or collective variables to evaluate how well they represent the mechanism, the committer, the potential mean force. And then we can actually get sufficiently accurate results that we can make very subtle changes to the molecule and actually see how things like the committer or the reactive current above the pathways, how those move around. So I think it's really quite a powerful technique. And I hope people are going to use it. So with that, I'll stop, and I'm happy to answer any kind of questions that people have.
Thank you, Professor Dina. That was a fascinating talk. I see there are a couple of questions in the chat box, but I think in the interest of time, we'll just allow one of them. So Jagannath, do you want to ask, or you want me to read the question?
I can ask actually. The question actually is that what we are trying to I try to understand is that this DGA that you try to propose, and if you look at the regular maximum likelihood based MSM, and which you are trying to point out the DGA is a generalization of this MSM. I'm just trying to figure out is that, let us say, for trip cage, which is fairly a model system, is it has it been tested that the rate that you get or the timescale separation that you get from regular MSM, how different it will be that you will get from DGA?
Yeah, so at least in our hands, with our data set, we find with MSMs and DGA, we get similar results, but neither of them are actually converged. We have to go to longer lag times than we can with the 30 nanosecond trajectories. And so we're actually not at a converged result. There is a data set from the Shaw group that is like a 200 or 300 microsecond data set. The problem with that data set is that it's just unbiased, a long unbiased simulation. And so there's actually only 12 or so folding, unfolding events in it. And so it actually has less statistical power than the short trajectory data set that we generated. We have analyzed that data set as well. Yeah, so but to answer your question, we really have to go to longer lag times to fully answer that question.
All right. Okay.
So thank you, Professor Dina. I think yeah, you can answer the questions that are there in the chat box. So in the interest of time, we move it to the